Introduction. Welcome, medical students, to today's lecture on the intricate processes of DNA replication. In this comprehensive script, we will delve into the fascinating world of molecular biology and explore the various components and mechanisms involved in the replication of our genetic material, DNA. Now, let us begin our exploration by examining the crucial role of DNA polymerase in the replication process. DNA polymerase is an enzyme responsible for catalyzing the synthesis of new DNA strands during replication. This essential protein possesses a remarkable ability to accurately copy existing DNA templates, ensuring the faithful transmission of genetic information to the next generation. The main function of DNA polymerase is to add nucleotides to the growing DNA chain in a sequence complementary to the template strand. This enzyme possesses an active site that specifically pairs incoming nucleotides with their complementary counterparts on the template strand, thus facilitating the formation of hydrogen bonds between the nucleotides. These bonds stabilize the newly synthesized DNA strand. Moreover, DNA polymerase has two distinct properties crucial for replication. First, it possesses 3 to 5 exonuclease activity, allowing it to proofread the newly synthesized DNA strand and correct any errors in nucleotide base pairing. Second, DNA polymerase can synthesize DNA only in the 5 to 3 direction, a characteristic dictated by the inherent polarity of DNA. As DNA replication initiates, a crucial structure known as the DNA replication fork is formed. The replication fork represents the region where the two DNA strands are separated and serve as templates for the synthesis of new DNA strands. This process occurs bidirectionally from the origin of replication, meaning that two replication forks are present in most organisms. The replication fork consists of two components, the leading strand and the lagging strand. Let us delve deeper into the synthesis of these strands. In DNA replication, the leading strand is synthesized continuously, while the lagging strand is synthesized in a segmented manner. This occurs due to the asymmetrical nature of DNA's antiparallel strands and the unidirectional activity of DNA polymerase. The leading strand is synthesized in the same direction as the replication fork movement. As the fork opens, DNA polymerase binds to the template strand and elongates the new complementary strand in a continuous manner, following the replication fork. On the other hand, the lagging strand is synthesized discontinuously. As the replication fork opens, DNA polymerase binds to the lagging strand template and synthesizes short DNA fragments called Okazaki fragments in the opposite direction of the replication fork movement. These fragments are then connected by DNA ligase, which we will discuss in more detail later. An essential enzyme in DNA replication is DNA helicase. It is responsible for unwinding the two DNA strands at the replication fork by breaking the hydrogen bonds between the base pairs. This unwinding process creates the single-stranded template necessary for DNA synthesis. DNA helicase moves along the DNA molecule, utilizing energy from ATP hydrolysis to separate the two strands in a process known as DNA unwinding. As it progresses, the replication fork expands, allowing access to the template strands by DNA polymerase. Another key player in DNA replication is primus. Primus is an RNA polymerase enzyme that synthesizes a short RNA primer on the DNA template strand. This RNA primer provides a starting point for DNA polymerase to initiate new DNA strand synthesis. Primus synthesizes the RNA primer by using the DNA template as a guide and incorporating complementary RNA nucleotides. Once the RNA primer is synthesized, DNA polymerase can bind to it and extend the primer, adding DNA nucleotides to form the new DNA strand. As mentioned earlier, the lagging strand is synthesized discontinuously, resulting in the formation of short DNA fragments called Okazaki fragments. These fragments are approximately 1,000 to 2,000 nucleotides long in most organisms, but their length can vary. Once the RNA primer is synthesized on the lagging strand, DNA polymerase extends it, synthesizing an Okazaki fragment. This process is repeated multiple times as the replication fork progresses, resulting in the formation of several Okazaki fragments on the lagging strand. DNA ligase plays a vital role in DNA replication by sealing the gaps between Okazaki fragments on the lagging strand. It catalyzes the formation of phosphodiester bonds between adjacent DNA fragments, creating a continuous DNA strand. After the RNA primers are removed by specialized enzymes, leaving gaps between Okazaki fragments, DNA ligase enters the scene. DNA ligase recognizes these gaps and catalyzes the formation of phosphodiester bonds, joining the adjacent DNA fragments and completing the synthesis of the lagging strand. Telomeres are specialized structures located at the ends of linear chromosomes. They consist of repetitive DNA sequences and play a crucial role in maintaining chromosome stability during DNA replication. Due to the nature of DNA replication, the conventional synthesis machinery fails to replicate the full length of linear chromosomes. As a result, telomeres act as protective caps, preventing the loss of genetic information by compensating for incomplete replication. To ensure the fidelity of DNA replication, organisms have developed intricate proofreading and error correction mechanisms. DNA polymerases possess 3 to 5 exonuclease activity, which allows them to proofread the newly synthesized DNA strands. If an incorrect nucleotide is added, the exonuclease activity enables DNA polymerase to remove the incorrect base and replace it with the correct one. This proofreading mechanism significantly reduces the occurrence of replication errors. Furthermore, specialized repair enzymes monitor the DNA replication process and identify any mismatches or errors missed by the proofreading activity of DNA polymerase. These repair mechanisms correct errors and ensure the accuracy of the replicated DNA. DNA replication origins are specific DNA sequences where the replication process initiates. A particular set of proteins recognizes and binds to these regions, forming a pre-replication complex. The pre-replication complex further recruits additional proteins, leading to the assembly of the replication machinery necessary for the initiation of DNA replication. Once assembled, the replication machinery unwinds the DNA helix and starts synthesizing new DNA strands bidirectionally from the replication origin. To tightly regulate DNA replication, an essential process called licensing occurs before the initiation of each replication cycle. During licensing, proteins bind to DNA replication origins and mark them as available for replication. This licensing process prevents multiple rounds of replication from occurring at the same origin simultaneously, ensuring that each section of DNA is replicated only once per cell division cycle. Strict control over DNA replication licensing is vital for maintaining genomic stability. Maintaining high replication fidelity is crucial for accurate transmission of genetic information from one generation to another. The fidelity of DNA replication depends on the precision of DNA polymerase in incorporating and proofreading nucleotides. Evolution has fine-tuned DNA polymerase to achieve an extremely high level of fidelity, with an error rate as low as 1 mistake per billion bases replicated. This remarkable accuracy allows organisms to maintain genetic stability and minimize the accumulation of mutations over multiple generations. In conclusion, DNA replication is a highly orchestrated process involving numerous enzymes, proteins, and molecular mechanisms. Key players, including DNA polymerase, helicase, primase, and ligase, work together to ensure the accurate synthesis of 2 new DNA strands during each replication cycle. The intricate coordination of leading and lagging strand synthesis, along with proofreading and error correction mechanisms, guarantees the fidelity of DNA replication. Replication origins and licensing further regulate the initiation of replication at specific sites, while telomeres protect chromosome stability. By understanding the intricacies of DNA replication, scientists strive to gain insights into diseases caused by replication errors and develop therapeutic interventions to ensure the integrity and faithful transmission of our genetic information.